Hello everyone, my name is Suwali from the University of Illinois. Today, we will discuss the effects of increasing a noble phytase in diet on digestibility of minerals and energy in pigs. Traditional diets for pigs in the US mainly contain corn and soybean meal. In most plant-based feed ingredients, including corn and soybean meal, significant amounts of phosphorus are phytate bound. The concentrations of phosphorus in corn and soybean meal are 0.26 and 0.81% respectively, but as mentioned earlier, about 81% of phosphorus is phytate bound in corn and is 47% in soybean meal. Phosphorus bound to phytate may be released by the enzyme phytase, but pigs secrete very limited quantities of phytase. Therefore, pigs are not able to release most of the phytate-bound phosphorus in feed ingredients, which is the reason they have low digestibility of phosphorus. This thus will bring negative impact on economics, and the excess excretion of phosphorus may increase environmental pollution. To increase the phosphorus utilization, several strategies may be used. Feed processing technology, including heat treatment or distillery processes, may increase phosphorus utilization. We can also use transgenic pigs that are able to secrete enough phytase in saliva to utilize phosphorus from phytate, or transgenic plants that contain low phytate phosphorus or high intrinsic phytase. However, the first two may not be the most used methods for increasing phosphorus utilization in the swine industry in the US. To increase phosphorus utilization in diets for pigs, calcium phosphates are used because digestibility of phosphorus in the feed phosphates are much higher than in plant-based feed ingredients. Lastly, exogenous phytase is commonly used in commercial diets to increase digestibility of phosphorus from phytate in plant-based feed ingredients, which reduces the need for feed phosphate in diets and also results in reduced excretion of phosphorus in the manure. Superdosing phytase in diets may also increase digestibility of nutrients such as amino acids and other minerals, even though they are not the substrates for phytase. Here are the possible mechanisms with exaggerating animations. Phytate is negatively charged in the gastrointestinal tract of pigs. Because of the negatively charged reactive sites on the phytate molecules, chelated mineral phytate or nutrient phytate compounds may precipitate in the intestinal tract. However, phytase hydrolyzes the ester bonds and thus releases the chelated nutrients, which results in increases in digestibility. Another hypothesis is that before nutrients get to bind to phytate molecules, phytase hydrolyzed the ester bonds to prevent them to chelate, which also increases digestibility of phosphorus and other nutrients. In this experiment, a novel E. coli phytase was used. This phytase starts the hydrolysis at position C6 of the inositol ring. Even though the phytase is non-coated, this phytase is heat-stable, and this phytase is also unique in that the production efficiency is higher than other phytase. The objective of this experiment was therefore to test the hypothesis that increasing phytase in corn soybean meal-based diets increases the apparent total tract digestibility, ATTD, of energy, the standardized total tract digestibility, STDD, of phosphorus, and the ATDD of minerals. For this experiment, 48 pigs with the initial body weight of 22.6 kilos were utilized, and they were fed 6 diets to have a total of 8 replicates per diet. All diets contained corn and soybean meal. The first diet was the positive control diet that contained phosphorus at the requirement for standardized total tract digestible phosphorus by 11 to 25 kilo pigs. The second diet was the negative control diet that was similar to the positive control diet, with the exception that no feed phosphates were used. 4 additional diets that were similar to the negative control diet were formulated to contain 250, 500, 1,000, or 2,000 units of phytase per kilogram of diet. Therefore, there were 5 negative control diets. Fecal samples were collected quantitatively for 4 days after 5 days of adaptation. Fecal samples were dried and ground to be analyzed for dry matter, gross energy, phosphorus, and other minerals. After feed intake, fecal excretion, and the digestibility values were calculated, data were analyzed using mixed procedure of ASAS. The model included diet as the fixed variable and group as the random variable. Contrast coefficients were used to compare the positive and negative control diets and linear and quadratic effects of increasing phytase in the negative control diets. Moving on with the results, let me set up the slides. In the x-axis, we have the positive control diets and the 5 negative control diets with increasing phytase from 0 to all the way up to 2,000. Here in the y-axis, we will have response parameters. And all the next graphs will have the same structure. The TTD of dry matter in the negative control diet containing no phytase was greater compared with the positive control diet. This can be explained with the diet formulation that the positive control diet contained more feed phosphate than the negative control diet, which may reduce dry matter digestibility. However, there was no effect of increasing levels of novel phytase on the ATD of dry matter in the negative control diets. We also determined the ATD of gross energy and digestible energy in diets. And the results indicated that the ATD of gross energy in the positive control diet was less than in the negative control diet when no phytase was used. The reason for the reduction can also be explained by the fact that feed phosphates contain no energy and the negative control diet does not contain any feed phosphate. Similar to the ATD of dry matter, we did not observe the effects of increasing phytase on the ATD of energy in the negative control diet. Because the ATD of energy increased, the concentration of digestible energy in the negative control diet was greater than in the positive control diet when no phytase was used. There was no effect of phytase on the concentration of digestible energy in the negative control diet. This slide shows the STTD of phosphorus in percent. The results from this experiment indicated that the STTD of phosphorus in the positive control diet was greater than in the negative control diet when the phytase was used. This observation demonstrated that use of feed phosphate in the positive control diet increased the digestibility of phosphorus compared with negative control diet. Increasing levels of phytase from 0 to 2,000 units quadratically increased the STTD of phosphorus in the negative control diet. This result indicated that the novel phytase was able to break down the ester bonds between phosphorus and the inositol ring of phytate and thus increased digestibility of phosphorus. This slide shows the ATD of calcium in percent and the ATD of calcium in the positive control diet was greater than in the negative control diet without phytase, which was also the result of using DCP in the positive control diet. Increasing levels of phytase from 0 to 2,000 units quadratically increased the ATD of calcium in the negative control diet, which was not a surprise to us. It is well known that phytase increases digestibility of calcium because use of phytase releases calcium from calcium phytate complex or prevents complex formation. Based on this, it is important that the release of both calcium and phosphorus by phytase should be considered in diet formulation. This slide shows the ATD of magnesium in percent. The ATD of magnesium was not different between the positive and negative control diets, but the ATD of magnesium increased linearly as phytase increased in the negative control diets. However, under the condition of this experiment, we did not observe any effects of dietary calcium and phosphorus or increasing phytase in the diet on the ATD of potassium, sodium, copper, iron, manganese, and zinc. It has not been conclusively demonstrated that phytase also increases digestibility of minerals other than phosphorus and calcium in diets fed to pigs, and inconsistent results among experiments have been reported. In this experiment, only digestibility of magnesium was increased by phytase, but we do not have any explanation why only magnesium was influenced. In conclusion, the ATD of dry matter and gross energy was lower in the positive control diet compared with the negative control diet because of the use of feed phosphate. The use of feed phosphate increased the digestibility of phosphorus and calcium in the positive control diet than in the negative control diet. Increasing phytase from 0 to 2000 units per kilogram of diet increased the digestibility of phosphorus, calcium, and magnesium. However, the ATD of energy, dry matter, and other minerals was not changed. With that, we would like to acknowledge Hanli International for their valuable inputs and financial support. Thank you for your interest in this presentation. If you enjoyed my presentation and you'd like to know more about pig nutrition, visit our website. You can find the website by Googling pig and Stein. Thank you for your attention.